So I mentioned that there are a couple ways that cytochrome P450 can influence drug response. It's not just drug elimination, right? So that's the bottom half here, metabolized to inactive drug. Remember, so if you're a poor metabolizer and you're taking a drug that's inactivated by your cytochrome P450, that means you're slow to metabolize the drug. That means you have an accumulation of the active drug in your system, and you're at risk for side effects there. You may require a lower dosage. In this case, an ultra-rapid metabolizer, somebody who has more copies of the enzyme, more functioning enzyme, they're metabolizing the drug much more quickly. That means in this case, where you're getting rid of the drug, eliminating it from your system, that the drug may not work well for you. You might have poor drug efficacy because the drug is being cleared before perhaps it can be active in your system, and you're at risk of therapeutic failure. You might need a higher dose of the drug. But in the situations at the top where you're taking a prodrug and metabolizing it to a drug, or you're taking a drug and metabolizing it to the active metabolite, things are kind of flipped. So that in this case, if you're a poor metabolizer and you're slow to metabolize the drug, that may result in poor efficacy for you because you're slowly making the product that is active in your body. So in this case, the PMs are at risk for reduced therapeutic efficacy, and they may need a larger drug dose. In this case, the ultra-rapid people who metabolize the drug very quickly, they're at risk for the overdose because now they're making more of the active product than a normal metabolizer would. So you have to be aware of what the situation is for the drug and your cytochrome P450 that you're dealing with in this instance as to what it means to be either a poor metabolizer or an ultra-rapid metabolizer, because it varies depending on the situation and the drug. There are several cytochrome P450s that are implicated in pharmacogenomic associations. This is a short list of some CYPs. Today I'm going to discuss in this segment a little bit about the top four, CYP2D6, 2C19, 2C9, and 3A5 in particular. CYP2D6 has been mentioned before in this and other segments. It's a very complicated gene. There are at least 140 different alleles. There are other SNPs across the gene where we don't really know the phenotype, and it hasn't been assigned to a particular haplotype. So there are more than 140 different combinations. There are many alleles that have either no or decreased activity. And this is a gene where we do have gene duplication events, and there can be more than one copy on a chromosome, in some cases, in one case noted up to 13 copies. And this can result in increased activity of the cytochrome P450. In this slide, just trying to show you how, what happens with the whole gene deletion, if you have particular polymorphisms, or if you have duplication, how it results in either increased metabolism or no metabolism. So you can look at this chart in a little bit more detail on your own. It just shows the complexity of the different levels from a UM down to a PM. And here I've highlighted the star 5, which again is the gene deletion for cytochrome P450. In fact, about 10% of Caucasians have either decreased or no activity of CYP2D6. And actually, in East Africans, up to 30% of people there have hyperactive CYP2D6 based on the gene duplication events. So it's somewhat population related. The frequency of these different alleles are different in different populations, but different star alleles can be found regardless, in an individual, regardless of the population. CYP2D6 is very important in pharmacogenomics because it is the phase 1 metabolizing enzyme for about a quarter of the drugs on the market. It metabolizes, there's a list, lots of different classes of drugs, a lot of antidepressants, beta blockers, as well as codeine, an example you've heard. It's also inhibited by a lot of other drugs. The expression of CYP2D6 is inhibited by many other drugs. And this can lead to a lot of drug-drug interactions that pop up in drugs that are metabolized by CYP2D6. Because so many of these drugs are inhibitors, taking one of these inhibitory drugs at the same time as a drug that uses CYP2D6 to metabolize it alters the phenotype, the appearance of the phenotype or the appearance of the function in that particular patient. So if you're a normal metabolizer of codeine, for example, when you take codeine, you metabolize it to morphine, the normal dose is the right dose. But if you're at the same time taking drugs that inhibit the expression or competitively bind CYP2D6, you have less CYP2D6 to metabolize the codeine to morphine. So you're not able to metabolize the same amount. And you're going to look phenotypically like a poor metabolizer of of codeine. You're going to look like your CYP2D6 has enzymes have lower function, when actually you're a normal metabolizer, but it's because of these inhibiting other drugs that are inhibitors of CYP2D6. So it's very important to keep that in mind when you're doing studies with pharmacogenomic studies with cytochrome P450s, and you have patients on multiple therapies. You have to take that into account. So there are a lot of known PGX associations with CYP2D6. There are CPIC dosing guidelines for at least 10 drugs right now, including codeine and antidepressants such as tricyclic acids and SSRIs. The Dutch Working Group for Pharmacogenetics has written guidelines on 25 drugs and the activity of CYP2D6. There are at least 45 FDA drug labels that mention CYP2D6 on them. So again, this is a gene that has a lot of known PGX associations, and a lot of it has been documented either in guidelines or on these drug labels. And if you want to learn more about the allele frequencies across different populations and the predicted phenotypes from diplotypes, from the star allele assignments, you can go to this link on PharmGKB. It will take you to one of the CPIC guidelines that has a supplement that you can download and has tables of frequency information and functional information. So now I'm going to talk about CYP2C19 for a couple minutes. CYP2C19 is not quite as diverse as CYP2D6. There's about 35 different alleles known for CYP2C19. One particular allele, star 17, is well known because it is a variant in the promoter region of the gene, which results in increased transcription of the gene. So you have more of the enzyme, so you have increased function. So this is in CYP2D6, when we talk about ultra-rapid metabolizers and increased function, we were talking about gene duplications primarily. You had more copies of the gene on your DNA, and that's why you had more functioning enzyme. In the case of CYP2C19, there's a particular SNP in the promoter region which can cause the same phenotype, an ultra-rapid phenotype if you're homozygous for that particular SNP, because you're making much more of the enzyme off of just the 2 copies that you have. There are at least 15 alleles in CYP2C19 which have decreased or no function. So again, being a poor metabolizer is not completely rare. There are many allele examples. For CYP2C19, there are also many dosing guidelines available from CPIC and DPWG, a little bit fewer than 2D6, 8 drugs. Have CPIC guidelines on them, including clopidogrel and also antidepressants. So antidepressants are influenced by both CYP2D6 and CYP2C19. And there's 12 drugs that the DPWG has written guidelines about regarding CYP2C19. There are 16 FDA drug labels that mention CYP2C19. And again, for more information about frequencies and mapping the star alleles to function, you can follow this link here to PharmGKB. There are no dosing guidelines currently about proton pump inhibitors, but you'll hear a lot about CYP2C19 and this class of drugs. And just to show you quickly, this is a proton pump inhibitor called omeprazole. This is a cartoon of the drug in a liver cell, and it's primarily metabolized through CYP2C19 into inactive forms. So in this case, CYP2C19 is clearing the proton pump inhibitor from the body. And if you have decreased functional alleles, like a star 2 or a star 3, you may have more of the drug in your body, and you might actually be getting better efficacy from the proton pump inhibitor, especially in cases with people who are taking the proton pump inhibitor for H. pylori infections to get rid of the H. pylori infection. There's some evidence in the literature that there's a greater chance of being able to eradicate the H. pylori infection with proton pump inhibitors if you're a poor metabolizer. And if you're an ultra-rapid metabolizer, you're homozygous for star 17, you may metabolize your proton pump inhibitors much more quickly before they have a chance to really take effect in your body, and you may not get as much relief from proton pump inhibitors. Okay, I'm going to talk just briefly about CYP2C9. Again, there's about 35 different alleles for CYP2C9, at least 5 of them with decreased or no function. There's probably many more. I'll show you in just a second. And CYP2C9 is an enzyme where it appears to have substrate-dependent function. So I'm going to show you what I mean by that. In this chart, we have some different star alleles divided up by functional status. And we have some of the tests, in vitro tests, with different substrates to see what the clearance is of the particular drug. And that seems to vary here depending on what the substrate is. So in this case, we have our normal star alleles with normal activity include star 1 and star 9. But you can see that for warfarin and a different drug, we have slightly different rates of clearing the drug. And so it's an interesting situation because while they're similar, the rates, they differ enough that you couldn't necessarily say with certainty across drugs which star alleles. There's some ambiguity about which star alleles would be poor metabolized, or maybe how much decreased function there is with a particular star allele. So the decreased activity alleles here, star 2 and star 3, known to have decreased activity. Star 6, star 15, star 25, known to have no activity with these particular drugs in these particular cases. But there's many more star alleles where they probably have decreased activity. And you can see that it varies. You can take a look at this on your own or through the readings assigned with this segment to see how it varies slightly depending on the substrate. I also wanted to mention that star 2 and star 3 are the alleles that are commonly looked for when studying CYP2C9, looking for decreased function. And in a different segment, I talked about warfarin and some of the tests that people use for calculating the warfarin dose, and that the star 2 and star 3 alleles are the alleles that are commonly looked at, but that those alleles are prevalent in certain populations and not as prevalent as in others. And these other star alleles down here that are possible decrease could be just as important to check for depending on the population that you're studying. So just don't forget to bear in mind that when you only look for a few star alleles, you're missing many more. So the other thing to note with CYP2C9 is that it has a much simpler star allele naming system. In these cases, most of the star alleles only have one variation or one amino acid change that really defines the star allele. There may be other small non-coding changes that travel along with these, but for CYP2C9, it's much easier to test one particular location and be able to define the star allele based on that one location. There's one or more where they have maybe a couple variants used to define the star allele. But most of the time, this is a straightforward gene to genotype. Again, there are dosing guidelines with CYP2C9. CPIC has a warfarin guideline and phenytoin. The DPWG has 7 drugs that they give guidelines for. These can all be found, by the way, on PharmGKB, all the guidelines. If you go to the link at the bottom, you'll be able to find all the guidelines as well. There's 4 FDA drug labels with the mention of CYP2C9, also all available on PharmGKB, just for you guys to note, all the ones that I've talked about today. And for more information, again, on allele frequencies and the predicted phenotypes from the star allele mappings, you can go to this link. So the last cytochrome P450 that I'm going to talk about today is CYP3A5. And this CYP is a little bit different than the other ones. So one, there's no 3D structure in the protein data bank, and all the other ones I had a little picture, the 3D molecule, the crystal structure of the enzyme. This one doesn't have one. They only have CYP3A4. The issue with CYP3A5 and CYP3A4 is that they're very highly homologous. They have high sequence homology, and they have overlapping substrate specificities. So a lot of times, it's very difficult to know how much of a drug is being metabolized by CYP3A4 by CYP3A5. And CYP3A4 is one of the most prevalent CYPs in the liver. Even so, variation in it doesn't seem to impact drug response that much. So that's something that's currently being studied to try to figure out why, even though CYP3A4 are some of the most abundant CYPs in the liver and they metabolize the greatest number of drugs, variation doesn't seem to influence it. And it's difficult to tease out sometimes 3A4 from 3A5. But there's one instance, at least one instance, where CYP3A5 seems to stand out. Tacrolimus is a widely used immunosuppressive medication. It has a narrow therapeutic index and large interpatient variability. It's used a lot to treat liver transplant patients. And in this case, variation in CYP3A5 does seem to make a phenotypic difference in these patients. So the thing to note here is that the STAR1 allele, which denotes a normal metabolizer or wild type, is actually rare in the Caucasian population. So currently, the common dosage for drugs like tacrolimus is actually a dosage for poor metabolizers, for STAR3s, because that's the prevalent genotype in the Caucasian population. And so the common dosage was based off of that. So interestingly, if you were a poor metabolizer, if you are STAR3 for CYP3A5, you actually would take the normal dose of tacrolimus. But if you are a STAR1, which is a normal metabolizer, you may need an increased dose than the standard of care for tacrolimus because you're metabolizing more of that than the poor metabolizers are. So I hope that makes sense. It's kind of an interesting case. And again, you can read more about this drug and the influence of CYP3A5 in the CPIC guideline, which is available on PharmGKB. I mentioned drug-drug interactions in the context of CYP2D6 earlier, but I just wanted to repeat that drug-drug interactions for any of these cytochrome P450s can make the genotype for one metabolizer status, your germline metabolizer status, look like a different metabolizer status because of inhibition, competition with other substrates, or inducers. So there are also, we talked a little bit about inhibitors who prevent the enzyme from acting at the normal level. Inducers can actually increase the amount of enzyme produced and make your metabolizer status faster than it would be ordinarily. And so there are some really great tables at Indiana. I have the website here. They have extensive tables for drug interactions with cytochrome P450s. 1A2, 2B6, all the main cytochrome P450s are listed. They have one table for substrates one table for inhibitors, and one table for inducers. So if you want to know more about that, I suggest you go take a look at those tables. They are something to always keep in mind whenever you're doing studies with these cytochrome P450s and you're looking at patients that are on multiple medications. I wanted to note the pediatrics issue with cytochrome P450s. It turns out that expression of cytochrome P450s varies throughout your lifetime. So a lot of the CYPs are not expressed in the fetal liver, or maybe they are only expressed maybe at the end of pregnancy. It depends on the cytochrome P450. Some of the expression is ramped up immediately after birth. Sometimes hours after birth, there'll be a ramp up in expression of particular cytochrome P450, but complete maturation of the expression of certain cytochrome P450s may not happen until adolescence. So the primary, for example, the primary cytochrome P450 expressed in fetal liver is CYP3A7 instead of CYP3A4, which we talked about earlier. So it gradually shifts from 3A7 to 3A4 during childhood. The fact that the same cytochrome P450s are not expressed at the same rates at given ages is a challenge to pharmacogenomics, and one of the reasons why drugs that are effective or safe in adults may not always be the case in children because of these differences in the metabolizing enzymes that are available in their bodies. So it's just something to note, and some of the more developed dosing guidelines will take that into account and be able to tell the reader whether or not the guidelines are appropriate for use in a pediatric setting as well as an adult setting. Otherwise, the assumption is these recommendations would be applicable to adults, but not necessarily pediatrics. A really great resource for cytochrome P450 information is this website. They call it a database, the nomenclature database. The website is listed, the URL is listed here, and they are the kind of authority on cytochrome P450 variation and the naming. They're responsible for the nomenclature, and they assign these star allele names to the different variations. So if you want to look into that more, how things are named and what specific variations designate what specific star alleles, this is a great resource to go to. I use it all the time. This is the home page for it, and then I'm showing you a screenshot for CYP2C19. It's not a very interactive website. It's kind of flat files. This is just a table for CYP2C19, but they do have every variation that comes along with a particular star allele listed. They have the reference for who submitted it to their database and so forth. And phenotypic changes, functional changes when known are also listed here. So it's a really good resource for cytochrome P450.